Hello and welcome, dear viewers, to the interesting topic on total impurity limits. And in this video, we are going to understand about what are the impurity specifications, what is impurity profiling, and basics of the impurities. Then also we will understand how the total impurity limits are given and how the specifications are designed for total impurities. So impurities is any component of the new drug product that is not the drug substance or an excipient in the drug product. So it is not the drug or any component of the drug product which we know that we have added. So the impurity profiling is required. The impurities might be organic, inorganic, residual solvents, and any other material which is not a component of the drug product, and it is not also a excipient. Impurity profile means a description of the identified and unidentified impurities present in the drug product. So impurity profiling means we are making a list of the impurities in the formulation, and we are giving the impurity names if these are known. Also, if these are unknown, then we can specify it, or we can make the limit of unknown impurities, and then at last we make a limit of total impurity. So that is impurity profile. Now what are the impurity thresholds? So how these impurities are controlled into the drug substances and drug product, that is based on the threshold limits, and these threshold limits are provided into the guidelines, and the main guideline for providing the impurity threshold is ICH-Q3. It involves three thresholds. First is reporting, second is identification, third is qualification. So you can remember by making a short form RIQ: reporting, identification, and qualification. So reporting is a limit above which the degradation product should be reported. So here the word is degradation product, and above this we have enlisted the term as impurity. So impurity might be process impurity, which is coming from the drug product processing, like intermediates or reagents or catalyst, or the impurity may be formed due to the degradation of the API inside the drug product or in the drug substance itself. So mainly impurity may be of two types: the degradant and the process impurities. So these impurity thresholds are for degradation impurities, and generally the process impurities are not considered because these are controlled by the purification techniques and purging techniques. Then also the process impurities are controlled into the drug substance specification. So reporting threshold is a limit above which the degradation product or impurity should be reported. Identification threshold is the limit above which the impurity or degradation product is required to be identified. That is, the its structure should be made known and a name should be given. Then qualification threshold, a limit above which the degradation product or impurity is required to be qualified. And qualification means making the data available for the toxicity and safety for that particular impurity. So qualification will require first identification of the impurity, then that impurity will be tested for the toxicity and safety, and if it is found safe, then the level at which it is safe, that will be regarded as a qualified level. Then coming to the next is the threshold values given into the guidelines, and the thresholds are based on the maximum daily dose. So maximum daily dose means a dose of the formulation or drug substance which is given within 24 hours to the patient, and based on that the reporting threshold, identification threshold and qualification threshold are given, and these thresholds are considered on the basis of MDD, and on the basis of MDD the threshold are given in the percentage and microgram per day or mg per day, whichever is the lower. Based on this threshold and based on the maximum daily dose, the reporting, identification and qualification limits can be identified. So impurities may be known and may be unknown. So known impurities means the chemical structure is known and name is there. Also sometimes or many times generally the known impurities will have a standard for studying the impurity, that is known as impurity standard. Then unknown, the chemical structure is not known. These are called as unknown impurities, or if the unknown impurity is coming at specified RRT, that time it is called as specified unidentified impurity, and the RRT limit is given or RRT information is given. So the impurity specification means it is giving the limit to each and every impurity, and also the specification is given to the total impurity. So many of the guidelines gives the information about the individual impurities that may be known, unknown, specified, identified or unidentified, or any specified impurity or any other individual impurity or max unknown impurity. But many of the professionals and the people who are dealing into the impurity profiling and specification, they have less understanding or they get confused or they have less uh, well known to the criteria for total impurities. So this video will guide the people or professionals for this, how to give the specifications for total impurities. Now the total impurities are the sum of all specified and unspecified impurities above the reporting threshold. So reporting threshold means the threshold at which or above which the impurity is required to be reported, and total impurity considers the sum or summation or total of all specified and unspecified impurities above the reporting threshold. Now if it is below the reporting threshold, it will be disregarded. So unless otherwise indicated, the same definition applies to the total impurities into the drug product monographs. So the total impurity for drug substance and drug product is similar. The drug product monographs may include a note that certain drug substances process related impurities identified by the relative retent enzyme should be included in the total impurities. And when this note is included, the total impurities should include all specified and unspecified impurities above the reporting threshold, apart from those or apart from these impurities which are the process related impurities. So if the impurities are process related, then these are not included, and if these process related impurities are also degradant impurities, for example, impurity A is a process related impurity. It is controlled into the drug substance specification, and if you are taking that product, taking that drug substance and making a product, and you find that though the impurity A is a process related impurity into the API, but it is getting increased on stability or shelf life, and it is a degradant, and you can understand that this impurity is a degradant, then these type of impurities are required to be included into the total impurities. Some monograph may state disregard any peak below certain value in chromatographic test. In this context, the disregard limit is the decision criteria, that is the numerical value for the user whether a peak response of an impurity is to be included or excluded in the total impurities. Typically, the disregard limit for substances covered by monograph is set in accordance with the reporting thresholds. When the limit of the total impurities is prescribed, a reporting threshold needs to be included into the test for related substances. And why this reporting threshold is required to be included? Because based on this, we can know which impurity is to be taken into the total impurity and which impurity can be disregarded. This threshold helps to establish the minimum sensitivity need for the analytical system used. This is very simple, that if you know the reporting thresholds, then you will know the minimum sensitivity requirements. Peak corresponding to the blank can also be disregarded, as well as other peaks that the monograph explicitly states are to be disregarded. So this is the requirement, and as per the monograph, we can consider the disregard limits and the disregard criteria. In other words, if the impurities, these impurities may be specified or unspecified. So if these impurities are above the disregard limit, they should be taken into account for the calculation of total impurities. Now the impurities may be process impurity or degradation impurity, and some impurities are both type of process impurities also, and they are degradant impurities also. So the impurity specification includes each specified identified degradation product, each specified but unidentified degradation product, then any unspecified degradation product with an acceptance criteria of not more than the identification threshold. So this identification threshold criteria is given to the unknown and unspecified impurity. Then it comes into the picture the total degradation product or total impurity limit. The acceptance criteria for total impurity limits, excluding significant human metabolites, generally should not exceed the summation of acceptance criteria for individual specified impurities, and these individual specified impurities may be again identified or unidentified. That means specified, unidentified, and unidentified impurities. Also it includes the max unknown impurity. So the simple definition of total impurity becomes not more than total acceptance criteria or total of the acceptance criteria for individual specified impurities. ICH M3R2 defines that the significant human metabolites are those that occur at exposures greater than 10% of the total drug related exposure. So this metabolite criteria is also there. If the impurity is a significant metabolite, then the criteria for specifying the limit may be more, because these are the significant human metabolites, and generally the metabolites are considered as qualified into the preclinical and clinical studies. So there will be a very important and informative video on impurity limit for significant human metabolites, and it will come very soon. Now the acceptance criteria for individual impurities that are also significant human metabolites should be considered separately. So why the separate requirement is there if the impurity is significant metabolite? Because generally if the metabolite is more than the 10% of the total drug exposure, that time it is considered as significant. And if it is significant metabolite, it may have exposure mm, more than the impurity limits specified into the guidelines like ICH Q3. So the sum of the all impurity limits, including those for significant metabolites, should not exceed the threshold that may compromise product potency, that is assay, through drug product expiry. For example, if the assay limit is 95 to 105 and the total impurity limit cannot be like not more than 6%. So if your assay at initial stage is 100% and after 6 months the impurity total impurity get increased up to 6%, then the assay will be 94%. And this is very simple, that your assay or potency of the drug product should not get compromised because of the total impurities. And that's why generally the total impurity should not exceed the percentage which will compromise the assay or potency. The example here I have shown a hypothetical example for your easy understanding. The drug product is having impurity A, B, C, D. These are the known impurities, and any individual impurity or max unknown impurity. So the limits are given here as per the MDD and as per the requirements of guidelines. So 0.2%, 0.2%, 0.2%, 0.2% for impurity A, B, C and D. And suppose the total daily exposure of the drug product is 6-7 mg or 5 mg. So the as per the ICH identification threshold, the limit for unknown impurity will be 0.4%. So as per the guideline, the total impurity limit should not exceed then the total summation or total of these all individual impurity specification limits. So 0.2 plus 0.2 plus 0.2 plus 0.2 plus 0.4 becomes 1.2%. And here this 1.2% is not affecting the assay value or potency value. Now this is the specification, and if you get the actual results like enlisted here, 0.01%, 0.02%, 0.02%, impurities D is not a much more degrading or not increasing, it is 0.1%, 0.3%. This maximum impurity is going up to the 0.3%, and the total becomes 0.45. But in the real time, you get the total impurity as 0.7%. Why? Because there are number of impurities which are also getting generated, and these are about the disregard limit. So these are taken into the consideration while calculating the total limit. And this is very practical example given here, and there is very limited information for this understanding. That's why I made a video on this. So real time example will give you the impurity percentage of 0.07%. So can you say that the product is failed? No, the product is not failed. The product will fail if it crosses the limit of 1.2%, and that will also vary on the case by case basis. The general considerations we will see now and we will discuss. So what will be the release and shelf life criteria for total impurities? If you make a specification for total impurities including or excluding the significant metabolite, and you make a specification for release and you make a similar specification for shelf life, but the shelf life testing may give you total impurities very less than the acceptance criteria set, or little bit higher than the acceptance criteria set as summation or total of the all specified impurities and the unknown impurities. Then at that time we should consider tightening or relaxing the impurity limits, and these are taken on case by case basis, and this differs product to product. Also, the storage conditions are required to be taken into consideration. The packs and pack configurations, the product strength are required to be considered and studied. And based on this all the data, then the impurity limits for total impurity may be tightened or may be relaxed. Also, this is a matter of the regulatory authorities, whether the regulatory authority permit this type of practice or not. Then tightening the total impurity limits are based on the stability data of EB batches, validation batches, commercial validation batches, or based on the real-time data or significant body of data after the commercial launch of the product. So that involves the commercial life cycle. That approach can be used, and based on that approach, total impurity limits can be tightened or relaxed. Then consideration of significant metabolite or metabolite impurities and total impurities. So if the product is showing a metabolite as an impurity or degradant, so that is required to be considered separately, and judiciously the impurity limit is required to be given to the impurity which is also a significant metabolite, so that sometimes the assay may get hampered if you consider the metabolite impurity into total impurity, and sometimes it may not get hampered. For example, your assay limit is 95 to 105 and the total impurity limit is 4%. So there are less chances for the effect on the potency. Now if this total impurity of 4% just I have mentioned contains 2% of the metabolite impurity and other 2% contains the summation of known and unknown impurities, so it will also not affect. So these are taken on case by case basis and differs product to product. Also, always there should be a strategy to minimize the impurity generation by careful evaluation and research activities, so that though the impurities are below the specified limits, we can save the public health by giving the stable product and the product with less impurities. So I hope this video will help you to understand the total impurity limits. Thanks for watching the video and keep watching the informative videos, as the Pharma Learning in Depth channel is providing the good understanding about the topics which are of a great interest in the interviews. Also, please do share to these videos to your friends, so that the professionals can improve their knowledge and understanding about the different topics. Thanks for watching the video.